Pharmapal.
Clinical applications of omics technology as a bioinformatic tool. Welcome back to our channel. Today, we're diving into the fascinating world of omics technology and its clinical applications as a powerful bioinformatics tool. So, what exactly does omics mean? Omics is a term that encompasses a range of scientific disciplines that study biological systems on a large scale. This includes genomics, which focuses on the complete set of DNA, proteomics, which studies proteins, and metabolomics, which looks at metabolites, the small molecules involved in metabolism. In the realm of healthcare, omics technologies are revolutionizing how we approach disease diagnosis and treatment. By analyzing the entire human genome, researchers can identify genetic variations that may predispose individuals to certain diseases. This is where bioinformatics comes into play. It's the computational backbone that helps us make sense of the vast amounts of data generated by these technologies. 1. Genomics. This is the study of an organism's complete set of DNA, including all of its genes. By sequencing genomes, scientists can pinpoint mutations that might lead to diseases. 2. Proteomics. Here, we focus on the entire set of proteins produced in an organism. Understanding how proteins interact and function can reveal insights into disease mechanisms and help identify biomarkers, substances that indicate the presence of a disease. 3. Metabolomics. This field examines the metabolites in biological samples. By analyzing these small molecules, we can gain insights into metabolic pathways and how they change in various diseases. The clinical applications of omics technology are vast. For instance, nutrigenomics, a branch of genomics, studies how food affects gene expression. This can lead to personalized nutrition plans that optimize health and manage diseases related to nutritional imbalances. Another exciting application is pharmacogenomics. This area focuses on how genetic variations affect individual responses to drugs. By tailoring medications based on a patient's genetic profile, we can enhance drug efficacy and minimize adverse effects. During the COVID-19 pandemic, omics technologies played a crucial role. Whole genome sequencing of the SARS-CoV-2 virus provided insights into its structure and behavior, aiding in vaccine development and treatment strategies. Moreover, integrating various omics data, known as multi-omics, allows researchers to create a more comprehensive picture of health and disease. This integrated approach can lead to innovative treatments and a better understanding of complex conditions like cancer, diabetes, and cardiovascular diseases. Next, let's discuss nutrigenomics. This is the study of how foods and their components influence gene expression. It plays a crucial role in nutritional optimization and can help manage severe diseases related to nutritional imbalances or deficiencies. Next, we have transcriptomics. This field focuses on the complete set of RNA transcripts produced by the genome. It aids in our molecular understanding of diseases, allows for disease profiling, and serves as a valuable diagnostic tool. Then there's proteomics, which involves the large-scale study of proteins produced in an organism. This branch is vital for disease management, identifying disease biomarkers, and serves as another diagnostic tool. Following that is metabolomics. This area studies metabolites within cells or organisms. It's essential for understanding disease pathophysiology, managing diseases, and identifying biomarkers. Lastly, we have aerogenomics, which combines traditional Ayurvedic concepts with modern genetics research. It focuses on personalized care and therapeutic management based on individual genetic profiles. Now, let's shift our focus to the evolution of omics technology milestones, from the early days of natural cytotoxic cells in the late 1960s to the groundbreaking CRISPR gene editing technology in 2012. Each of these advancements has contributed to our understanding of biological systems and paved the way for future innovations in healthcare and medicine. Finally, let's take a look at the workflow for integrated omics. This integrated approach combines various omics data, like genomics, transcriptomics, proteomics, and metabolomics, to provide a comprehensive view of biological processes. The workflow starts with data collection and progresses through various stages, including data analysis and interpretation, ultimately leading to insights that can inform clinical applications and therapeutic strategies. Next, let's explore genomics, the study of an organism's entire genome, which is essentially its complete set of DNA, including all of its genes. This technology has advanced significantly, allowing us to analyze whole genomes quickly and affordably. In fact, it's revolutionizing how we approach medical diagnostics and treatments. Now, let's talk about some key applications of genomics in medicine. First up, we have gene discovery and medicinal biology. Genomics technology is crucial for identifying genetic abnormalities. Scientists can now pinpoint specific genes responsible for genetic diseases. Just think about it, over 4,000 genetic disorders have been identified in recent years, a huge leap from just 50 in the 1990s. Next, genomics helps us understand common diseases. By analyzing genetic factors, we can gain insights into the progression of diseases like hypertension, cancer, and neurological disorders. This knowledge is essential for developing effective treatments. Now, let's move on to pharmacogenetics and targeted therapy. This is where genomics gets really interesting. By understanding an individual's genetic makeup, doctors can determine how well a specific drug will work for them. This means more effective treatments with fewer side effects. For instance, in cancer treatment, we can identify genetic mutations that drive the disease and select drugs that specifically target those mutations. Another critical application is prenatal diagnosis and testing. With advancements in genomic technology, we can now conduct non-invasive prenatal testing. This means we can identify potential genetic disorders in a fetus early on, allowing for better planning and care. Genomics also plays a vital role in understanding infectious diseases. By sequencing the genomes of pathogens, scientists can uncover how these microorganisms work and spread, which is crucial for developing effective treatments and vaccines. And let's not forget about personalized medicine. This approach tailors medical treatment to the individual characteristics of each patient, based on their unique genetic profile. It's all about customizing healthcare to improve outcomes. Lastly, we have gene therapy and genome editing. These cutting-edge techniques allow us to modify genes to treat or prevent diseases. Imagine being able to correct genetic defects at their source. Hey everyone! Welcome back to our channel. Today, we're diving into an exciting and rapidly evolving field known as nutrigenomics. So, what exactly is nutrigenomics? Let's break it down together. Nutrigenomics is the study of how our genes interact with the foods we eat. It explores the relationship between our diet and gene expression. In simpler terms, it looks at how different nutrients can influence our genes and, ultimately, our health. Imagine your body as a complex machine. Just like a machine needs the right fuel to operate efficiently, our bodies require specific nutrients to function optimally. Nutrigenomics helps us understand which nutrients are best suited for our unique genetic makeup. So, how does nutrigenomics work? It's all about understanding the gene-nutrient interactions. Our genes can affect how we metabolize different foods, and conversely, the nutrients we consume can influence gene expression. For example, certain genes might make you more susceptible to conditions like obesity or diabetes if you consume a diet high in sugars and fats. On the flip side, a balanced diet rich in fruits, vegetables, and whole grains can help mitigate these risks. Now, let's talk about some practical applications of nutrigenomics. 1. Personalized nutrition. With nutrigenomics, we can create personalized diet plans tailored to an individual's genetic profile. This means that what works for one person might not work for another. It's all about finding the right balance for you. 2. Disease prevention. By understanding how certain foods affect our genes, we can take proactive steps to prevent chronic diseases. For instance, if you have a genetic predisposition to heart disease, nutrigenomics can guide you towards a heart-healthy diet. 3. Nutritional interventions. Researchers are conducting large-scale nutritional intervention studies to validate the concepts of nutrigenomics. These studies can help identify specific dietary changes that could benefit individuals based on their genetic makeup. Next stop is transcriptomics, specifically focusing on its applications in comparative transcriptomics. So, what exactly is transcriptomics? In simple terms, transcriptomics is the study of the complete set of RNA transcripts produced by the genome under specific circumstances. This includes messenger RNA, or mRNA, which carries the genetic information needed for protein synthesis. Now, why is this important? While understanding RNA expression levels can provide invaluable insights into how genes respond to various stimuli, such as environmental changes, disease states, or developmental processes. Comparative transcriptomics takes this a step further. It allows scientists to compare RNA expression profiles across different organisms, conditions, or time points. This comparison can help identify conserved and divergent gene expression patterns, shedding light on evolutionary processes and functional adaptations. For instance, let's say we're studying a specific plant species. By comparing its RNA transcripts with those of a closely related species, we can uncover which genes are responsible for traits like drought resistance or disease tolerance. This knowledge can be crucial for agricultural advancements and crop improvement. Now, how do researchers perform comparative transcriptomics? They often use high-throughput technologies, such as microarrays or RNA sequencing. These methods allow for the simultaneous analysis of thousands of genes, providing a comprehensive view of gene expression. Once the data is collected, bioinformatics tools come into play. These tools help analyze the vast amounts of data generated, enabling scientists to identify significant differences in gene expression between the samples they're studying. Next, let's talk about the typical steps involved in a proteomics study. It starts with purification, where proteins are isolated from a sample. Next, we move to quantification, which is all about measuring the amount of each protein present. Then comes characterization. This is where we determine the properties of the proteins, such as their structure and function. To achieve this, we use various chromatography-based techniques like mass spectrometry and gel-based approaches. Mass spectrometry, for instance, allows us to analyze the mass-to-charge ratio of ions, helping us identify and quantify proteins. Other techniques include Edman sequencing, Western blotting, and protein microarrays. Each of these methods has its unique strengths and applications, allowing researchers to gain insights into protein behavior and interactions. Now, let's shift gears and discuss metabolomics. Metabolomics is the study of metabolites, which are the small molecules produced during metabolism. This field helps us understand the biochemical processes happening within living organisms. Next, let's examine a typical metabolomics and lipidomics workflow. The workflow begins with a biological question. This is crucial because it guides the entire study. From there, we move on to study design and validation. This step ensures that our approach is robust and minimizes potential biases. After that, we collect samples. This is a critical stage. Timing and consistency are key to avoiding variability in our results. Once we have our samples, they need to be prepared for analysis. This involves various steps, including metabolite extraction and quality control sample preparation. Once everything is ready, we move to data acquisition. Here, we utilize techniques like LC-MS, liquid chromatography mass spectrometry, and NMR spectroscopy to gather our data. After acquiring the data, we enter the data processing phase. This includes peak detection, data normalization, and compound annotation. Essentially, we're cleaning and organizing our data to make it usable. Next, we analyze the data using statistical tests to identify any significant differences between samples. Finally, we integrate our findings into a biological context using pathway analysis and visualization tools. This helps us understand how our results relate to biological processes. So, why are proteomics and metabolomics so important? Both fields provide invaluable insights into disease mechanisms, drug development, and personalized medicine. By understanding the proteins and metabolites in our bodies, researchers can identify new biomarkers for diseases, develop targeted therapies, and improve patient outcomes. Next is lipomics, also known as lipidomics, and aerogenomics. These areas are at the cutting edge of biological research and have some exciting implications for health and medicine. First up, let's talk about lipomics. This branch of omics technology focuses on lipids, which are essential components of cellular membranes. Think of lipids as the building blocks that help form the structure of our cells. They play a crucial role in various biological processes, including energy storage and signaling. Lipidomics involves comprehensive studies of lipids to understand their relationships and functions within biological systems. researchers use high-throughput techniques. These are methods that allow scientists to analyze large amounts of data quickly to explore lipid profiles in different conditions, such as disease states. This can help us identify how lipid imbalances might contribute to health issues. For instance, improper lipid metabolism can lead to several diseases, including obesity, diabetes, and cardiovascular diseases. By understanding lipidomics, we can develop better diagnostic tools and targeted therapies, especially in drug discovery and development. Now, let's shift gears and explore genomics. This innovative field combines the ancient wisdom of Ayurveda with modern genomic science. Ayurveda is a traditional system of medicine that emphasizes balance in the body through the concept of three dashas: Vata, Pitta, and Kapha. Each dasha represents different physical and emotional characteristics. Genomics seeks to understand how these dashas influence our genetic makeup and overall health. By integrating genomic data with Ayurvedic principles, researchers aim to create personalized treatment plans that consider an individual's unique genetic profile and lifestyle. This approach can lead to more effective preventative medicine and wellness strategies. For example, if a person's genetic makeup indicates a predisposition to a certain condition, genomics can help tailor dietary and lifestyle recommendations that align with their specific needs. In essence, genomics forges a new link between traditional and modern medicine, promoting a patient-centered approach rather than a one-size-fits-all solution. It's about understanding the whole person, body, mind, and spirit. Pharmacogenomics and toxicogenomics, two cutting-edge fields that are revolutionizing how we understand medicine and toxicity. First off, what exactly is pharmacogenomics? Well, it's the study of how our genes affect the way we respond to medications. Imagine a world where your doctor knows exactly which drug will work best for you based on your genetic makeup. That's the promise of pharmacogenomics. It helps tailor treatments to individuals, ensuring safer and more effective therapies. Now, let's shift gears to toxicogenomics. This field examines how our genes respond to toxic substances, like chemicals or drugs. By understanding the genetic factors that influence toxicity, researchers can better predict adverse reactions and develop safer products. Now, you might be wondering about the relationship between these two fields. That brings us to Figure 7.11, which illustrates the connection between toxicogenetics and toxicogenomics. Toxicogenetics focuses on the genetic variations that affect individual responses to toxicants, while toxicogenomics looks at the broader picture, how entire gene expression patterns change in response to toxic exposure. Together, they provide a comprehensive view of how our genetics influence our reactions to drugs and toxins. Next, let's talk about the pros and cons of different omics approaches, starting with genomics. Pros: it helps identify single nucleotide polymorphisms, or SNPs, which can provide valuable insights for early disease diagnosis and treatment. Cons, however, it can be challenging to predict the final biological effects of DNA analysis alone, due to factors like post-transcriptional modifications. Now, onto transcriptomics. Pros: this approach identifies major pathways involved in drug responses and toxicity, with high reproducibility across labs. Cons, but it has limitations too. Data can be insufficient due to post-translational modifications, and changes in the transcriptome may not always lead to changes in end products. Next is metabolomics. Pros: it has fewer metabolites than genes or proteins, making data interpretation easier. It's also useful for finding genetic markers for diseases like cancer. Cons, on the downside, metabolomics can be expensive and may yield different outcomes due to post-translational modifications. Finally, we have proteomics. Pros: this technique allows for the analysis of low-abundance proteins in complex samples. Cons, however, it can be costly and may not be sensitive enough for all proteins, limiting its applicability. So, as you can see, each omics approach has its strengths and weaknesses. The integration of these technologies is crucial for advancing our understanding of drug efficacy and toxicity, ultimately leading to better patient care. Thanks for tuning in. If you found this video informative, don't forget to like, subscribe, and hit that notification bell for more insights into the world of genomics and personalized medicine. Until next time, stay curious.
Pharmapal.